Good afternoon. All right. So during our last discussion, the last time that we had our synchronous class, I failed to record the start of the at the start of the meeting. So I started only recording our lecture when we reached item number 20, I think. So I will just edit later and merge this portion to the previous lecture that we had. Okay. So I will just try to elaborate further your answers from items 1 to 19. Okay. So for number 1, elaborate the statement. Live fish has sterile muscle. Fish, while it is still living, its muscle is sterile, and that is true, no? But the moment it dies, and especially when there are ruptures in the skin, in the viscera, microorganisms can easily penetrate the different tissues and organs of fish. So that's why it's very important to handle your fish properly and always keep it at low temperature. Okay. For item number 2, discuss what rigor mortis is and what causes it to occur. So basically the answer here is rigor mortis is the stiffening of the muscle of the fish shortly after death. And note that the stiffening starts at the tail of the fish. Okay. And it is caused by a succession of chemical changes, and such changes include accumulation of lactic acid and a drop in pH. But take note, no, diba, when the fish dies, the circulation or blood circulation rich in oxygen also ceases. So by then, from aerobic respiration, the process now for it to continuously produce um, energy or ATP is to go into anaerobic glycolysis. In that process, fewer molecules of ATP are produced. I'm sorry, is produced, and what do you call this? Fewer molecules of ATP is produced, and at the same time, one of the byproducts of anaerobic glycolysis is lactic acid. And as the process proceeds, eventually there will be accumulation of lactic acid that will result to the drop of pH of fish muscle. So that is why pH is also an indication in monitoring the freshness of fish, no? So pag medyo matagal na siyang namatay, mas acidic na yung fish. Normally, the pH of fish is around 7 to 7.5, but once it's nearing spoilage, it drops to as low as 6.2 to and below. Pero hindi naman siya masyadong acidic to the point na nagiging 4, hindi siya umabot ng ganun. Unless pinrocess mo siya and nag-add ka ng acids. Okay? For number 3, why is it important to know the onset of rigor mortis in this one is more important, especially to those who are handling the fish or to fish processors, because knowing the onset of rigor mortis will provide you knowledge, no, on how are you going to handle the fish. So for example, you are planning to fillet it. So if it is in rigor stage, you know that it is difficult to fillet, and you will yield a very low yield. In addition, it is also important, no, to know the timing of rigor mortis, because the texture of the fish is also affected kung ano siyang stage, no. So the one assigned to this item has indicated here that if the fish is cooked pre-rigor, the texture is very soft and pasty, while if it is in rigor, no, the texture is tough but not dry. Okay, so it is really important to know the status of the fish. What are the strategies in controlling the onset of rigor mortis? Okay, so there are a couple of strategies that can be applied to control the onset of rigor mortis, which first include the setting of fish at chilled conditions. It is important that the fish be placed in a cooled environment, no, before it undergoes rigor mortis, and it should remain in that condition until it pass rigor mortis. Kasi it will have a better quality if nag-rigor siya na cold ang temperature ng fish. Discuss why pH of fish muscle decreases post-mortem. So yun yung explanation kanina, no, na once mag-stop yung supply ng oxygen-rich blood sa system ng fish, it will stop aerobic respiration, and it will now go into anaerobic glycolysis. So for item number 6, agree or disagree with the statement and explain your answer. Production of energy in fish muscle ceases upon death of fish. So the answer is here is agree. Now, agree with the statement. Pero actually, you have to disagree. Kasi as I've mentioned earlier, no, even though patay na yung fish, it can still produce some molecules of ATP. But yun nga, hindi siya kasing dami ng aerobic respiration. However, still, we can claim that it still produces energy. Okay? So yun yun. Actually, the answer is, yung explanation niya dito is correct, except that dapat nag-disagree siya. Yun lang. Okay, so you know already if I say true or false, ganito. So alam niyo na yung sagot. So here, the production of energy in fish continues even after death of fish, as you have mentioned above, via anaerobic glycolysis. So in this process, however, lower amounts of ATP are produced and acidic products are accumulated. Okay? Pag naubos na yung glucose, that's the time na talagang magsa-stop na yung production ng energy. Okay? Number 7, how can you distinguish a pre-rigor from a post-rigor fish? So tama, it is through sensory evaluation. Yung mga ito kasi, comments ko na yan. Ayan. So, when you press the fish, kaya nga yung mga mother natin, kapag namamalengke, they are really touching the fish and check. No, they are touching and checking the fish if naiwan doon yung finger marks nila sa body ng fish. Ayan. So, hindi lang natin masyadong na-observe yun. I don't know if you're also going with your mother sa market, but yun yung purpose nila. Pag ano daw kasi, nagmamark yung finger mo doon, if it leaves a finger marks sa muscle or sa skin, that would mean na partially spoiled or stale na talaga yung fish. Okay? For number 8, discuss a laboratory procedure on how you will be able to determine the onset and duration of rigor mortis. Bakit rigor mortem? Rigor mortis. Ayan. In catfish. And kaya ako pinili yung catfish kasi, 'di ba? Catfish remains alive even you have already removed it from its aquatic environment for several hours. Okay? So you can really monitor its death, the time it dies and the time it will start to go into rigor mortis. So kumbaga, pag namatay siya, you record that time, no, na namatay, and then bantayan mo ilang hours after death, the fish starts to stiffen, and then you also monitor how long it remains in that position na matigas siya, and anong oras din siya nag-undergo ng resolution of rigor, yung state na nag-soften siya. And then, yun na yung ano mo, pwede mo nang gawa ng report yun. So, before, during the time when we still had the face-to-face Fish 160 activities, no, I gave my students what? Small ice buckets, and then they put their fish inside, and they make a setup wherein once the fish stiffens, no, pwedeng pag gumanon yung fish, kasi limp siya, halimbawa meron siyang ditong wood, nakaganon siya, limp siya. But pag nag-stiffen siya, gaganon. So, mababantayan nila dapat kung kailan gumanon yung fish, nanigas. And what they're doing is, they make bit-bit the ice bucket from one class to another until such time na makomplete nila yung experiment. How would they know na completed na yung experiment? Pag nag-stiffen na yung fish, na-monitor nila yung time, and the time na nagbalik siya, na soft na siya ulit. So, that's the end of their experiment. So, sometimes, it's taking them around 2 to 3 days bago matapos, kasi makunat yung buhay ng catfish. Matagal din siya minsan mamatay, and matagal din siya mag-undergo ng rigor mortis. But we're doing that experiment at room temperature kasi minsan napupunta pa sila ng AS na gano'n. Hindi nila pwedeng i-stop basta-basta yung experiment, and hindi nila kayang magbitbit ng ice all the time. The next item is number 9, discuss the biochemical aspect of the stiffening of fish muscle. So it's given here that after the fish dies, the brain control and blood circulation in its body cease. Okay, and this will result in the inability to process energy and transport various material essential in living cells. Living. Okay, so calcium here plays a very important role, no, in the stiffening of the muscle. So in the live fish, muscular contraction starts when a nervous impulse sets off a release of calcium from the sarcoplasmic reticulum. So it is the brain that tells the system of the fish to release calcium. So yun, may impulse, and then there will be release of calcium from the sarcoplasmic reticulum to the myofibrils. And then when calcium concentration increases, no, the ATPase, the enzyme ATPase is activated, and that will now lead to the sliding of actin and myosin filaments. So this causes a release of energy, no, to be used as a contractile energy, making the actin filament slide in between the myosin filaments. So later I will show you a very short video around 17 seconds, no, on how actin and myosin slide. Excuse me. Or how actin filaments slide over the myosin filaments. Okay, when the reaction is reversed, as in the case of dead fish, calcium molecule is pumped back, no, and the contractile ATPase activity stops, and the filaments are allowed to slip passively past each other, and the muscle is relaxed. So wala nang energy that will really control the sliding of actin between the myosin filaments. So that's why it's starting to be limp and soft. yung muscles. Okay, so ATP is one of the most important components of muscle contraction and functioning both as a fuel for contraction and plasticizer. So allow me to stop sharing this screen and let me share another screen now to show to visually show to you the sliding of actin and myosin filaments. So consider that this is the Z line, no? Z line in the muscle. Parang ano to siya? It's a representation of a sarcomere, no? That contains the actin and myosin filaments between the Z line. So ito yung green ang ating actin and ito yung orange ang ating myosin. So it says here that sliding filament theory, under sliding filament theory, that myosin filaments are use energy from ATP to walk along the actin filaments with their cross bridge. So again, this is your myosin and this is your the brown thing are your actin. Okay. So this pulls the actin kasi nagaganon siya. Oh, ito yung yung actin mo. Parang yung myosin mo tinutulak-tulak niya ito. Tinutulak niya ito yung actin mo para come in closer sila. So dahil nagiging closer itong dalawang, nagmi-meet yung dalawang actin, it causes contraction. So ganun yun. So this pulls the actin filaments closer together, causing the contraction or shortening of the muscle. Ito yun.
A biochemical ratchet acts when muscle contracts. Projections on the myosin filament, shown in red,
Ayan.
attach themselves successively along the spiral actin filament, which is represented in brown.
Ayan. So that's very short, but that's a very short video, but it's very useful, right? Sige, balik tayo dun sa ating document, Word document. Where are you now? Ayan. So, ang role ng calcium molecule dito is to activate the ATPase enzyme. Okay? So, yun. Next is, why is it important for a processor to determine the status of the fish? So yun yung briefly yung sinabi ko kanina, that it is important for the processor to know the status of the fish because of course, if it's in pre-rigor state, alam niya na talagang kakamatay lang ng fish and it's still very, very, very fresh. Okay? If it's in a rigor state, the processor knows that it should be placed in a cold environment or frozen condition or chilled condition in order to maintain its good quality. And also, the processor should not, as much as possible, fillet the fish because it is very stiff and the fillet quality will be very poor pag finilay niya na nasa rigor stage. Okay? Briefly, the processor would know how to handle the fish properly if he knows the quality status of the fish. So compare rigor mortis in exhausted fatty fish versus exhausted lean fish. So the answer, I am the one who gave an answer here. Some species take longer than others to go into rigor because of their differences in chemical composition. So take note, fish with high lipid content goes into rigor earlier than low-fat fish species. So if i-compare itong fatty and lean, kasi pareho naman silang exhausted. So, between fatty and lean, mas mauunang mag-go into rigor yung fatty fish. Okay? While kung hindi naman sila parehong exhausted, so exhausted fish goes into rigor faster or earlier than a relaxed fish. Relaxed. Meron ba ganun? Okay. Okay, so for number 12, compare rigor mortis in fish collected quickly from areas with abundant food and the same species of fish that have struggled prior to catch. So kumbaga, between the two, Yung first is mas relaxed, mas busog, mas madaming energy reserve. Itong number 2, it has struggled, meaning exhausted siya, depleted, almost depleting na yung energy reserves niya. And gutom kasi hindi naman galing sa versus the one that's coming from an area with abundant food. Okay? So which one will go into rigor earlier? Itong species B. Okay? Yan. So for number 13, discuss ATP degradation in fish. So ATP degradation in fish is catalyzed by several enzymes. Okay? So, briefly, ATP, pag na-degrade siya, goes into ADP, adenosine diphosphate, and then IMP, inosine monophosphate. But take note, inosine monophosphate here is very important degradation byproduct of ATP because it gives the fish its characteristic sweet fish or characteristic sweet fish flavor. So while IMP is a desirable byproduct, inosine and hypoxanthine naman are the undesirable ones kasi especially the hypoxanthine, it causes a bitter taste doon sa spoiled na fish. Okay. Each of these degradation products in fish, no, ATP degradation products can actually be measured. Ayan, so ngayon pwede na tayong mag HPLC, no, to measure the amount of IMP present sa sample, ATP, AMP, inosine, and all of those sorts. Okay. Explain the phases of quality changes in fish as shown in the graph below. So, ayan, I think this has been explained here naman, no? Ito yung graph. So, we have the phase 1, phase 2, phase 3, and phase 4. Mas inexplain dito yung freshness, no, ng fish. And take note that at day 12, nagkaroon na dito ng rejection of the sample. So, ang phase 1, as explained here, is the stage, no, where the fish is still very fresh and has a sweet and delicate taste. While in phase 2, still the fish remains acceptable. No, however, it has lost its characteristic odor and taste. Pero wala pa siyang off flavors. Okay? However, in phase 3, spoilage is already starting and there are a range of unpleasant smell that is produced, no? And that would depend on the species. Kapag kapag very fatty ang fish mo, so tendency magiging rancid siya because of the high amounts of polyunsaturated fatty acids of fish sample. Okay, and during phase 4, the fish is already spoiled and putrid. Okay. Discuss bacterial enzymatic degradation of TMAO. How do you measure TMAO degradation? Okay, again, so how do you discuss or discuss bacterial enzymatic degradation of TMAO? So first, we have to know that TMAO, no, is a nitrogenous compound that functions as an osmoregulator. No, and usually those fishes that live in the deeper parts of the oceans are the ones that have high amounts of TMAO. It's a naturally occurring ano din siya, nitrogenous compound, especially to those that requires really osmoregulations in their existence doon sa ating ocean. Ayan. So, TMAO degradation also leads to development of unpleasant fishy odor. So this degradation can actually be caused by an enzyme or enzymatic, or can also be due to bacteria or bacterial degradation process. So the degradation caused by bacteria can be caused by several facultative anaerobic bacterial species like the Shewonella, no, in which they reduce the MAO to trimethylamine, and yun yung nagbibigay ng unpleasant fishy odor sa ating fish, no? Take note also that bacterial spoilage, no, or bacterial degradation of the MAO takes place at chilled condition, while enzymatic spoilage takes place when you keep your fish at 0 to negative 30 degrees Celsius. Ayun, so mas mababa yung sa enzymatic. Okay, everything naman has been explained by the one assigned to this number. No, dito. Ayan. So, yung second question kasi, how do you measure MAO degradation? So, meron tayong tinatawag na, yung sa lab, kapag face-to-face tayo, you can perform an activity in which you can measure the amount of TVBN. Ayan, or total volatile base nitrogen na present sa sample. Kasi mga volatiles yung kailangan mong i-measure. So, how do you do that? You have you'll be using a Conway microdiffusion method, wherein meron kang parang Conway plate, no? And then dun sa plate, meron siyang outer ring and meron siyang inner ring. So, you incubate your sample with the solution here that has an indicator na kapag capture niya yung mga volatile bases dito, madidissolve dito yung mga volatile bases. So, yon. And so, that's how Conway microdiffusion work. Hindi ito, cup lang ito ng C2. Meron ka pa dito parang another bigger cup na nakapatong siya dyan. So, yun yung iyong setup, magiging setup. So, that's how you measure TMAO degradation. So other than that, meron pa tayong mga spectrophotometry, and pwede rin, may mga HPLC tayo. Yan yung mga medyo high-tech na technique. But during our time, we are using Conway microdiffusion method, and it's fun. Ayun. Next is, what is meant by SSO and why is it important to identify them in fishery products? So SSO stands for specific spoilage organism. So these are the organisms that are dominating and capable of producing metabolites that can affect the sensory properties of the product and causing consumers to reject the product. So yun yung magandang definition, very brief at very direct na definition ng SSO. So why is it important to identify them in fishery products? Because they are the ones that they're the ones that causes the spoilage of our products. So kailangan lang naman natin talagang makilala yung mga kalaban natin, no, in processing. Ayan. So take note also that they are specialized microorganisms that can quickly multiply during preservation and develop of odors and flavors that is linked to spoilage and also to spoilage metabolites. Ayan. So explain why the total count is higher than the SSO in the graph below. So obviously, kasi total, no? Not all the bacteria that can be present in your product are SSO. Hindi lahat SSO. Part of the total population, yun lang yung SSO. So it's what do you say? How do you say that? The total count is always higher than your SSO. Kaya naman ganun yung itsura ng graph natin. Saan ba yung graph? Ayan. So the total count is always higher than your SSO. Pwedeng mag-equal sila kapag lahat ng SSO mo, lahat ng bacteria na present sa sample are specific spoilage organisms. Pero kasi meron din tayong mga other organisms that does not necessarily cause spoilage. Present lang sila dun sa product. Ayun. Next is Tama ba to? Na-answer ba natin? Okay. Next is, discuss histamine formation in fish. Histamine formation results when a histamine-producing bacteria that are naturally present in the different parts of the fish synthesize the histidine decarboxylase enzymes during spoilage and fermentation. Okay. Please take note that, sorry guys inuubo kasi ako. Histamine formation is only prevalent in species that has naturally high amounts of histidine. At tandaan nyo na kapag walang histidine or mababa yung histidine doon sa sample na yon, hindi rin magkakaroon ng mataas na histamine formation. Ayan. However, histamine formation, kapag mataas yung level of histidine sa sample, the formation of histamine is triggered by high temperature, no? And yun, high temperature, and pag sinubject mo sa matagal na time na nasa ganung temperature yung fish mo. So, time and temperature abuse ang tawag doon. Okay? And ano to siya? It's a very important consideration in processing itong histamine that's being produced in the product kasi it causes allergic reaction, and some or the effects in some individuals can be fatal. Okay? Lalo na kapag masyadong mataas yung histamine content. Okay? So discuss how fishery products for export to the EU markets are tested for histamine. So, and describe the basis of product attention. So ito, in-explain, yung first part is well explained by the one assigned to this number. So merong mga analytical methods. However, there is a limit for histamine content for specific products. Look at this one. So for fishery products that have undergone enzyme maturation, around 200 to 400 milligrams of histamine per kilogram of sample yung allowed. And for fish sauce, it's around 400 milligrams of 5 to 400 milligrams per kilogram of sample. So dapat, as much as possible, the histamine content of your product should always be below 200 milligrams per kilogram of your sample. So meron tayo ditong N equals 9, small n equals 9, small c equals 2, M equals 100, and capital M equals 200 milligrams. So the small n here is the number of samples taken. And then C here is the number of parang critical samples. And M, small m here, sets the limit. And the capital M is the final na histamine content of your product. So I added here a parang elaboration nun, no? So in a batch, 9 samples will be taken, and then the mean value must be below the 100 ppm na limit, and no 2 samples should have a value more than 100 ppm. So ito sinabi ko na parang critical limit, critical number of samples na hindi dapat magkaroon ng dalawang sample na more than 100 ppm. But if there is one sample, isa lang na sample mo is 200 ppm and up, rejected na yung product. Pag may dalawang samples ka na 100 plus but below 200 yung histamine content, nare-reject din yung product. Kapag ang total na histamine content na nakuha doon sa 9 samples is above 100 ppm, it will be rejected din yung sample. So ganun, medyo mas busisi itong histamine content, no, for product detention. And so I guess we're done with this one. So I think that will be all for this lecture, and see you in your exam. This file available, or it's already available actually in the LMS. So okay lang. Kaya lang mamimiss nila yung mga sharing natin. Hay nako, bakit kasi hindi siya nag-auto record? Sige. Anyway, going back to item number 20, no? You just have to always remember that fish or lipids from fish is or are healthier lipids, no, or fats as compared with lipids that can be found from beef and pork. Okay, and the reason why fish lipids are more susceptible to oxidation kasi nga fish lipids have higher amounts of polyunsaturated fatty acids, and PUFAs are more susceptible to degradation kasi very unstable sila. So since they are unstable, they are prone to oxidation. Ayun. So yun yung pinaka answer to this question. Okay? Parang affected talaga ako dun sa fact na hindi na record yung first 19 items. All right. So number 21, explain how hydrolysis results in off odor and off flavor in fish. Ayan. So hydrolysis is a reaction with the involvement of water as one of the reactants. So due to enzymatic action, ATP, which is the main component of fish muscle nucleotide, undergoes hydrolysis. So it reacts with water producing ADP and one inorganic phosphate. So ADP further degrades into adenosine monophosphate and then to IMP and then to hypoxanthine. So IMP is considered to be responsible for the fresh flavor of fish. Yun yung sinabi ko kanina na sweet flavor. And its degradation to hypoxanthine gives the fish an off flavor because of the accumulation of the hypoxanthine in fish muscle. And it results to bitter taste and loss of fresh fish flavor. Ayan. So note also that hydrolysis is linked with lipid oxidation, and fats and oils are hydrolyzed by moisture, and this leads to hydrolytic rancidity, causing unpleasant flavor and aroma of the product. I guess ito na yung aking comment, no? Ayan, that hydrolysis is linked to lipid oxidation, and lipid oxidation results to off odor and unpleasant flavor. Yun. 22, discuss why temperate water fishes have different bacterial composition from those collected from warm waters. Obviously, we have different types of microorganisms, right? So we have the mesophilic, we have the psychophiles, we have the thermophilic. Yun. So the composition of or the microbial flora in its environment also varies with temperature. So iba yung magiging composition ng temperate water versus the temperate and tropical waters. Ayun. So obviously the tropical waters will have higher number of microorganisms as compared with temperate waters. Yeah. So ang remarks niya, hi, I strongly agree with your ideas that the different bacterial composition of temperate water fishes and warm water fishes depends on their living environment where they are caught. Oo, tama naman yun. Very simple actually ang question na ito. Okay? Next, discuss lipid oxidation in fish that results in off odor and off flavor. So lipids are a group of compound soluble fats generally found in plants and animals, whereas in this discussion, fish, hindi ko siya maintindihan. Anyway, fats, as we all know, are the primary energy storage of animals and are used for a considerable amount of movement during the periods of energy and food deficiency. So to answer this question, fish lipids, no? Oxidation of lipids found in fish produces secondary byproducts in the form of ketones and aldehydes. And these ketones and aldehydes are the ones responsible in the of odor and of flavor in spoiling fish. Okay? So medyo mahaba yung sagot. Basta yun na yung brief answer to the question. Okay? Next is explain how enzymes cause spoilage in fish. Okay, who wants to volunteer to answer this one? Yes, Ed Rose, please go ahead.
So enzymes cause fish spoilage in terms of autolysis. So autolysis or self-digestion happens due to the enzymatic degradation that results in cell damage of the fish. This process occurs as soon as the fish undergoes rigor mortis, causing the release of an autolytic enzyme that degrades the cell components like proteins, fats, etc., thus causing the physical and chemical changes to fish, including its appearance, flavor, and smell. The physical degradation of fish is visible since there are a lot of indications that can be seen by the humans, such as belly bursting, bumps, and spots. The changes of the flavor is a result of the ATP conversion to hypoxanthine and the decomposition of fish. Furthermore, the foul smell caused by autolysis is due to the degradation of muscle protein into amino acids, ammonia, CO2, amine, etc. is due to proteinase enzyme, which is one of the known autolytic enzymes. This triggers the production of secondary metabolites to produce indoles, ketol, etc., causes the foul smell of the fish. According to FAO, autolysis has different effects for different fish species when they undergo rigor mortis. In some, enzymatic changes relate to the freshness of fish and are not related to the microbiological activity. There are also cases in which the enzymatic changes precede and predominate the spoilage. However, there are times in which autolysis contributes to the overall quality loss of the fish freshness and relates to the microbial activities.
Okay, thank you, Ed Rose. So, briefly, no, actually very self-explanatory yung answer provided in this particular item. And it's correct, okay? So, hindi na ako masyado nag-comment dito. But briefly, no, enzymes attack fish tissues or fish muscles once merong mga physical damages sa fish. Example of that is belly bursting, nag-burst yung belly, and then mga bruises, no? And that will also lead to the rupture of the cells, and also ma-release na ngayon yung enzymes that will eventually cause autolysis in the fish. So, kasi yung mga enzymes, some of them are packed in the lysozymes. So, pag may mga damages doon sa fish tissues and sa organs or any part of the fish, they will be triggered to, or they will be released. So, they can already be activated and they will act on the tissues and cells. Okay, so mag-lead na sila to autodigestion of the fish. Okay, natapos na natin. Kaya lang, kaka-record ko lang. Okay. Sige. So, there are how many of us here? Okay, so stop share ko muna ito. So, I wanna see all of you. Wow, tayo ay 6 lamang. Okay. So, what's your takeaway in this discussion? Brief answer lang to my question. Ang takeaway nyo. Ed Rose.
Hello, ma'am.
Hi again.
So, ang akong takeaway sa muning discussion, ma'am, is that simple things like transporting and handling of fish has great impacts sa post-mortem changes sa fish and sa quality sang fish meat itself. Tapos nga it does not end lang bala ma'am sa like for example kung sa tandaan kung like hindi na fresh ang fish muna na sa dayon kay gali may undergo pa siya different chemical and biological processes to para magmuna ang iya state.
Okay. Very good. How about you, Faye Dubla? Duwag liaton, Faye.
Hi, ma'am. So, yung takeaway ko for this session po is very interesting yung mga processes na nangyayari even after death ng fish. And especially for the item that I was assigned, which is the which disagree or agree with the statement. Um, actually, I've also first disagreed with the statement. Yung
O pala yun, guilty ka.
ma'am. Ano po, medyo tricky yung question pero thank you for the clarification, ma'am, sa session po na to. Na ayun, ma'am, nasa kasama na po yung answer dun sa may na-cite ko pong reference, but still yung answer ko is nag-agree ako. Pero I disagreed first, pero change ko na agree kasi na-confuse po ako. Pero at least, ma'am, thank you for the clarification and na-understand ko na po yung concept behind the statement.
Okay, very good. Actually, maganda na kayo yung nag-provide ng answer and then diniscuss natin ngayon. I'm sure kung anong item kayo na-assign, you will always remember that item and that concept, and I'm sure then na hindi nyo siya makakalimutan for life kasi na-assign sa inyo yun. Teka lang, meron akong istorbo ditong bata. Say hi to my students. Hi, I'm Kalisi. Okay. Okay, you have your phone. Okay, go na. Okay, bye. Diyos ko, sorry ah. Okay. So, I'm happy to hear what you've said, Thea. Okay. Next, let's hear from another Thea. Ay, another Faye pala, Thea. Okay, si Faye pala yun kanina. So, ito si Thea Faye. Hagulo.
Medyo na-confused ka rin sa name. But my takeaway for this afternoon discussion is that nag-delve deeper kita on the process.
Hindi ko masyadong naririnig. Sorry.
Audible na po ba, ma'am?
Sige.
My takeaway for this afternoon's discussion is that nag-delve deeper pa on the concept of rigor mortis. Tapos yung mga little details like sa histamine information, yung sa SSO, yung sa graph, mas nagkaroon ako na nag-shed light pa siya on a further understood po, ma'am. Nadidinig po ba ako, ma'am?
Okay, oo.
So that will be all.
Okay. Yun din ba yung item na na-assign sa'yo, yung graph?
No, ma'am. Yun po yung ni-remarks ko lang.
Okay. So, diba, you are assigned to answer one item and then you need to comment dun sa another item. So, may sure kayo na meron kayong dalawang fully understood na concepts. Kasi you can't give a remarks kapag hindi mo rin siya alam. Diba? Ang saya talaga ng quiz na to. Actually, I was triggered to give you a quiz that time kasi nga hindi niyo ako sinipot ng previous meeting, pero I think meron ding mali dun sa LMS na hindi siya nakita. So, I am not blaming you na hindi kayo naka-attend nun. Kaya lang, syempre, I waited for more than 30 minutes. Wala akong estudyante, what happened? Okay, nag-guilty ako dun. Okay. Sige. And next, let's hear from Era.
Hello, ma'am.
Hello.
So, yung takeaway ko for this session, parang mas na-understand natin kung bakit nangyayari yan physically or externally sa fish if alam din natin kung anong nangyayari sa loob niya. Parang ganun po. And
Okay.
also, very important na alam mo yung ano ng state ng fish mo kasi mag-a-affect yan sa after pag-consume ng tao and also yung further processes na i-undergo pa niya. Yes, yun po.
Okay. Thank you. Next, si Irish.
Hello, ma'am. So in this discussion, it's really enlightening to know the mechanisms behind processes involved in fish handling and processing. I was particularly interested in the bacterial and enzymatic degradation of the MAO and yung mga steps na kailangan kong gawin para ma-secure ko na yung isda na bibilhin ko sa wet market is of high quality po. So, dapat kasasamahan ko na palagi si mama sa pagbili.
Samahan mo na siya. Kasi tuturuan mo siya na ganito dapat. Ayan. Okay. So, di ba, ang discussion natin in Module 1 is, ang title ng Module 1 natin is Fish as a Perishable Commodity. So, as mentioned, as we did the discussion doon sa answers ng quiz, we are reminded to always keep our fish cool, no? So, to limit or minimize bacterial action, bacterial degradation, and enzymatic degradation as well. So, yun. And doon naman sa Module 2, yung title nun is Fish as a Food Commodity. We know that fish is a very good source of protein, high quality proteins, and in addition to that, it contains or it's a good source also of healthy lipids or fats. And however, because of those polyunsaturated fatty acids, kasi yun yung mga healthy lipids natin, those PUFAs are very unstable and they are very prone to lipid oxidation. That's why we have to also keep our fish in good storage condition so as to minimize or retard the oxidation process in fish. So yun yun. And the third discussion is on, nakalimutan ko yung third na title ng module. Ano ba yung title nun? Pre and post-mortem changes. So we had an extensive discussion on pre and post-mortem changes in fish. So we know how to distinguish a fish that's in pre-rigor, in rigor, and in post-rigor state. Okay, for that particular topic, you have to take time to check my lecture that I have already uploaded in the LMS, no, lecture on module 3 part 1. Okay, so I don't know if we still have time, but meron pa tayong 20 minutes, I think. We just have to look at this slide on agents of fish spoilage. So I'm sure naman na nakaka-relate na kayo dito kasi some of the topics here were already tackled dun sa quiz, no? Na diniscuss natin. But ayan, i-run through lang natin. So you have to take note, no, that there are several agents in fish spoilage, and this includes the enzymes, the chemical agents, no, that's responsible in the oxidation of lipids, and also the microorganisms. Most importantly, the microorganisms, no. So we have the natural microbiota or the natural microflora of fish, and we also have the SSO or the spoilage microbiota. So, 'di ba? Pag sinabing SSO, alam niyo na agad 'yon, dapat. So, teka, i-move ko yung ating mga picture kasi naka-ano siya. I-move ko lang somewhere. So, yon. What other causes of fish spoilage or the things or factors that may contribute to fish spoilage are the following: physical damage, improper handling and storage, and of course, temperature abuse. Ayan. So this slide summarizes the events, no, in fish after death. So there will be ano tawag dito? Magsa-stop yung blood circulation in fish, and which also means that the blood supply in the fish muscle and tissues will also stop or cease, no? So from aerobic glycolysis, the pathway in the production of ATP will now shift to anaerobic pathway, which would lead to the accumulation of lactic acid, no? The ATP levels will fall or mag-decrease. There will be accumulation of lactic acid, and that will result to a pH level in fish tissues close to acidic or around 6, no? And there will be activation of enzymes at that particular pH. So the fish will now go into rigor mortis, and then after some time, it will go back to its limp and soft state, which is known as the resolution of rigor. And at that moment, there will be attack of microorganisms and also attack of enzymes, no, in the form of autolysis. And then those will produce metabolites that will further hasten the degradation or the spoilage of fish, because more and more microorganisms will utilize those metabolites in their growth, and they will now produce products that will cause the off flavor and off odor in fish. So yun yung magiging that's the time na magiging spoiled na, totally spoiled ang ating fish. So for the first one, enzymes, no? So these are substances, no, biological in origin, which catalyzes the biological reactions in fish, and they are naturally present in food, and could also be produced by microbes, which affects the texture attributes of fish. So pwede na yung microorganism will just attack the fish tissues and muscles, no? But they can also produce enzymes that will also hasten the spoilage of fish. So later you will know that in particular TMAO degradation can either be, as we mentioned earlier, can either be bacterial in nature or enzymatic in nature. Diba? Do you remember the TMAO reductase and the TMAO waste enzymes? So yon. So this is the process or yon process in nucleotide degradation or ATP degradation. And these are the enzymes, no? There are 7 enzymes that's involved in the degradation of ATP. So ang ATP will be degraded from ATP to ADP, AMP to IMP, and then inosine and hypoxanthine. Ayan. So as I have mentioned earlier, IMP is responsible in the fresh fresh and sweet flavor of fish, while the inosine and hypoxanthine are the ones that are responsible in the bitter taste of fish or of spoiled fish. Ayan. The production of IMP, no, is quick, very quick, kaya mabilis din yung pagko-convert niya into bitter taste. So mahirap din i-determine kailan magiging sweet. How long will the fish stay sweet? Ba para tayong tao, sweet. Stay sweet as you are. How long siya magsistay na sweet, no? Before it will go into bitter. Hala, yung fish natin, sweet muna bago maging bitter. Ano ba yan? Okay. Ayan, ito naman yung chemical formula of histidine, amino acid histidine, and your histamine. So it is the histidine decarboxylase enzyme that's responsible in the formation of histamine. And as what we've mentioned earlier, ang histamine is only formed in some species of fish, no? Only in those species that has initially high histidine content. All right? And the improper handling and storage of fish leads to or hastens the formation of histamine. And again, take note that histamine, no, once it is formed, it can no longer be eliminated from the product because histamine is heat stable. So kahit i-cook mo pa yung product mo, pag meron na siyang histamine, it can no longer be eliminated. It will remain makate, and it will still cause allergic reactions to sensitive individuals. So ganon. So the best way to prevent histamine formation is to properly handle and store your raw material or your fish. Okay? Next is TMAO degradation. So yun yung na-mention natin kanina, no? That it involves both the bacterial degradation acted on by bacteria and also by the enzyme TMAOase. So take note that bacterial spoilage, no, is what takes place at chilled condition, while the enzymatic takes place at frozen condition. So for the enzymatic, ang end product is the formaldehyde, in which it causes the toughening of the fish tissue or fish muscle. So later we will discuss once we talk about low temperature storage of fish. We will discuss also bakit at low temperature storage mas nagtatafen yung fish natin. Bakit nagiging dry and mas nagiging tough. Okay? And also, note that the end product of bacterial spoilage or bacterial degradation or TMAO degradation caused by bacteria, nagkakagulo-gulo na yung aking statement. The end product is DMA, just like this TMAO degradation caused by enzymes, and also the monomethylamine. So, ito yung difference ng dalawang processes. Next, ayan, wala lang, can be enzymatic or bacterial. The proteolytic enzyme. So, we have to briefly discuss, no, the cathepsins, no, they are the acid proteases that's packed in the lysozymes. So in living tissues or in living fish, no, hindi lang naman kasi siya nag-exist sa fish, in all living organisms, it's present, no. So they are inactive in living tissue and becomes active once released into the cell juices. So, in living tissue, lysosomal proteases are believed to be responsible for protein breakdown at the sites of injury. So, yon, kaya siya tinatawag na autolytic enzyme, itong cathepsin, okay? Ang second enzyme is the calpains, no? It is also known as the calcium activated factor. Ayan, associated with fish muscle autolysis and is found in meats, fin fishes, and crustaceans. So, it is responsible in the postmortem autolysis of meat through digestion of the Z-line proteins of the myofibril. So, if you're going to closely examine a myofibril, no, or yung muscle fibrils, no, you will find the Z lines, yung mga dark lines, no, that separates the sarcomere, no. And this calpains is the one responsible in the digestion of these Z lines. So imagine if itong dark lines here will be digested, so maseseparate mo na ngayon yung mga sarcomeres. Ayan, magka-cut na siya dito. So that's how crucial the function of calpains is, no? So in crustacean muscle, calpains are associated with molt-induced structural changes. Do you know what molt is? Molting? Sa shrimp, pag nagso-soften yung nagso-soft shell yung yung shrimp, most likely it undergoes molting. So anybody who can briefly describe what molting process is in shrimp, or in shrimp, for example? Anybody? Na-mention nyo, na-discuss nyo na ba sa sa inyong aqua classes yung molting? So molting is the shredding of the exoskeleton of crustaceans. So in the case of shrimp, parang i-shed nila yung kanilang shell, no? So pag nag-shed sila ng kanilang shell, it's an indication that they are growing. Kasi kumaga masikip na sila dun sa shell nila, i-shed nila yon, and then they will form a new shell. But during the time na nagmo-molt sila, they are very prone to anong term doon? Predation ba yun? Nakakainin sila posible nung kanilang kaparehong shrimp. May ganun na stage yung shrimp na when they undergo molting, they have to hide kasi pwede silang kainin kasi nagso-soft shell sila. Pwede silang kainin ng pareho nilang shrimp. Carnivorous din yung shrimp eh. It also happens in the crabs, no? Nagsished din ng shell yung mga crabs. So, pag may binibili kayong shrimp na soft, meaning kaka-molt lang. Ano ngayon yung nagko-cause nung hardening nung shell? After nilang mag-shred nung shell nila, 'di ba, soft shell sila, parang skin lang nila yung meron. And then eventually, after a while, they will again develop a shell. So, importante sa sa shrimp and some other crustaceans yung high amount of calcium carbonate in the water for them to be able to form again their hard shell. Ayun. Bakit ako nag-aquaculture? Ayan. Okay. In contrast naman sa fish, yung calpains is responsible in the digestion of myosin. 'Di ba kanina sa video mayroon tayong actin and myosin. So if na-digest na yung myosin, so yun, it will also lead to fish degradation. The third enzyme is the collagenases, no? It's the one that causes is the one responsible in the digestion of your collagen. And it causes gaping or breakdown of the myotome during long-term storage on ice or short-term storage at high temperature. Ayan. So in summary, these are the enzymes that are involved in the quality changes in chilled fishes. So ito yung enzyme. Take note, ito yung substrate nila. And then ito yung effect. And these are the prevention strategies. Okay, nakikita niyo yung cursor ko? Alright, so teka, i-check ko din yung time. Baka masyado tayong nag-o-overtime. Ito yung enzymes, no? So take note na sa TMAO natin, na TMAO demethylase, yung effect niya is formaldehyde. And yung prevention strategy is the storage of fish below -30. So hindi pa enough yung storage at negative 20. Yung household refrigerator natin kasi it's at negative 18 to negative 20. So hindi enough na i-keep lang natin yung fish natin at household ref. That's why yung mga nanay natin ayaw pa rin mag-stock ng isda kahit frozen siya. Kasi hindi enough yon to really keep your fish fresh. Kailangan at much lower temperature pa. Okay, you just studied this table, no? You have to note na ang calpains, they attack the myofibrillar proteins and it causes softening, no? And then yung collagenases, they attack, or yung substrate nila is the connective tissues and also causes softening and gaping of tissue. So yon. And then yung chymotrypsin and trypsin, these are your proteases, no? It causes belly bursting. And note na ang problem increases with freezing, no thawing, or long-term chill storage. So we will discuss more about chymotrypsin and trypsin when we discuss low temperature storage later on. Okay. Chemical, ito, lipid oxidation and hydrolysis. No, so lipid oxidation and hydrolysis takes place, no, could take place in a fish simultaneously. So again, the oxidation of fish oils yields rancid odors and taste, no, because of the secondary byproducts, which are the ketones and aldehydes. Ayan, ito pala siya. Ito yung mga secondary auto-oxidation products. Yan. This includes the ketones, alcohols, hydrocarbons. Ayan. So ang lipid oxidation, I think, is discussed in more detailed sa sa lecture na na-upload ko sa LMS. Okay, the third agent are your microorganisms. So what are the manifestations of bacterial spoilage? When you see visible growth, especially sa molds, no? Pag sa dried fish, mas obvious na makikita natin yung molds. Ayan, there are gas production in your products, slime formation, may mga ma-observe kayong off odors and flavors, no? So that's the manifestation of bacterial spoilage in your product. So fish is a very good substrate, no, for bacterial growth. So ayan, there are specific and non-specific contamination of the live animal from the environment and of products during processing, and growth conditions for microorganisms due to specific intrinsic and extrinsic factors. So take note that the intrinsic factors that contributes to bacterial growth are the following. No, the poikilothermic nature of fish, the high postmortem pH in the fish, fish flesh, the presence of large amounts of non-protein nitrogen. And also, the extrinsic factors includes the temperature, AW, microbial interactions, etc. So yung pag sinasabing intrinsic, it's natural to the fish, no? Kung ano yung present sa fish, while the extrinsic are yun nga, mga temperature, water activity, that is not naturally present in the fish. Syempre hindi naman ano yun, hindi siya yung microorganisms, hindi siya pinaka-part ng fish, 'di ba? All right, the normal flora of fish and shellfish, normal microflora of fish and shellfish. Ito 'yon. So the range is around 10 raised to 2 to 10 raised to 7 colony forming unit per square centimeters on the skin surface, and around 10 raised to 3 to 10 raised to 9 colony forming unit per square centimeters on the intestine and gills. So why is it mataas yung sa intestines and gills? Obviously, because they have, alam naman natin na the intestines and gills harbors large amount of microorganisms. So yon, ito yung mga dominant microorganisms for fishes from temperate waters. Ito naman yung sa warm waters. Of course, we have the psychrotrophs, the psychrophiles, and the mesophiles. For the coastal and the estuarine, we have the cholerae, the parahemolyticus, the Clostridium, and the enteric bacteria. Bakit madami yung enteric bacteria? Obviously, kasi nasa coastal. Okay? Hindi na natin kailangan i-elaborate ito. Okay? And for aquaculture organisms, mga Vibrio species yung common dyan. Okay? So, ito yung SSOs, no? Spoilage organisms are the specific producers of metabolites responsible for the off-odors and flavors associated with fish spoilage. So, kung marine temperate water fish yung na-store in ice, you know that the SSO is the Shewanella species. And if ice tropical freshwater fish siya, Pseudomonas yung yung SSO, and Aeromonas pag aerobically stored ang freshwater fish. So I think I have a table on this one. Okay na to, skip na natin kasi na-explain na yan kanina. So ito yon. These are the specific spoilage microorganisms or bacteria, and these are the spoilage compounds. Why is it important to know the spoilage compounds? Kasi you can measure these compounds, no? And you will also be able to determine or link this with your IDs of bacteria present in the product. So, yon. Ito yung mga spoilage compounds niya. So, I am sure you are familiar with TMA, that's trimethylamine, yung mga short abbreviations ng mga compounds. You can find them here, okay? Kapag mga amines yung compounds, most likely yung effect nun is, ang manifestation nun sa spoiled fish is ammoniacal odor and flavor. Kasi this would lead to the production of ammonia. Ayan. Ito naman yung mga microorganisms classified according to their rate of spoilage activity. So, pag high, yan, mga Pseudomonas. Pag moderate, so we have these microorganisms. And pag low to specific, may mga specific condition requirements sila. Ito yung mga Aerobacter, Lactobacillus, Flavobacterium, and Micrococcus and others. Right? So this slide shows the breakdown of amino acids by the action of bacteria. So pag amino acids, we know that proteins yung natin, yung substrate, no, proteins and amino acids. So these are the or the enzymes responsible for the breakdown of amines of amino acids. So may involved na deaminase and also the carboxylase, no, that leads to the production of ammonia. Yung end product is the ammonia. So ano yung result, no? Ang result niyan, ang obvious sensory manifestation for this is the odor and flavor. Of odor in particular, kasi nga ammoniacal odor yung magiging obvious na ma-observe natin sa products. Okay, I guess this is the last slide for this particular discussion. So I think you are now ready to take the exam. Ayan. So that's all for today. Thank you for attending up to the end of this session. Thank you for staying. Ayan.